Okay, let's get started. I'm glad to see you this morning, and we will start the next block of topics. It will be about tissues of the internal environment, and exactly about blood. This is what you should know very well. You should um, remember these four types of tissues, and you should remember this information during all the course of histology. This is the most important information that you should never forget. We have four types of tissues. First, epithelial tissue. Second, tissues of internal environment, which we will discuss next. uh, Nervous tissue and muscle tissue. Four types of tissues. And epithelial tissues we did study in previous blocks of topics, and the colloquium 1 is about epithelial types of tissues, and now we start second type, tissues of internal environment. What is most important when you learn tissues is their classification. So, tissues of internal environment include blood and lymph, tissues with specific properties, which are reticular, adipose, and pigment tissue. Next, embryonic tissues, which have two subtypes, mucose and the mesenchyme. Also, we have connective tissues proper, which are loose fibrous connective tissue and dense fibrous connective tissue. And we have also skeletal tissues, which are cartilage and bone tissues. So this is the classification of tissues of internal environment. All these tissues have common distinctive properties, some specific features common for them all, and these features include that these tissues develop all from mesenchyme. Also, in these tissues, cells lie separate from one another. It means that we have a lot of extracellular matrix in all such tissues. If you compare these tissues with epithelial tissues, where we have no or almost no extracellular matrix, here in all these tissues we have a lot of extracellular matrix. So we start with blood, which includes plasma and formed elements. So, organization of blood system. Blood system includes blood itself and organs of hemopoiesis and immune defense. The word hemopoiesis means production of blood. Hemo means blood, poiesis means production, creating of blood. Immune defense is about immune defense. So, functions of blood include, first, transport function. Blood transports gases, oxygen and CO2 gas, and thus performing function of respiratory. Also, blood transports nutrients and performs trophic function. Also, blood carries some biological active substances and performs regulatory function. And through blood some wastes are removed with the help of kidneys next, which make urine from blood plasma, and so blood performs excretory function. Second function, maintenance of homeostasis, which include osmotic equilibrium. It means the specific concentration of ions. Also acid-base balance, which means the pH level, which should be constant. And also body temperature also stays almost constant with the help of blood. And third function of blood is immune defense, which include innate or non-specific immune responses, blood coagulation process, which includes clotting of blood when the blood vessels are damaged, and also adaptive immunity, cell-mediated and humoral. specific immunity against specific bacteria and viruses. This is performed with the help of cells of blood. Functions of organs of hematopoiesis and immune defense include: first, renewal of circulating formed elements. It means that some of these organs make new formed elements of blood, and in some of them, aged formed elements of blood die. Second function, maintenance of immune homeostasis. It's about immunity system, because these organs perform immunity defense, and these organs produce cells of immune defense. Defense against microorganisms and foreign particles, immune surveillance over own cells of the body. Defense against microorganisms means defense against bacteria, viruses, and their toxins. Immune surveillance over own cells of the body means that the cells of immune system walk constantly through our body with the help of blood vessels and lymphatic vessels, and watch over all the cells of our body, because some of the cells may become tumor cells and cause cancer. If this happens with some cells, the cells of immune defense will find such bad cells and kill them. And also temporary blood storage is also additional function of some of the organs of immune defense. So, composition of blood. Blood is a tissue, and as any tissue, it includes cells and their derivatives and extracellular matrix. The cells and derivatives of cells in blood are called formed elements of blood, and they make about 40–45% of volume of blood. Extracellular matrix of blood is liquid, and it is called plasma, blood plasma, and it is about 55–60% of volume. We have the so-called hematocrit, HCT, hematocrit, which shows the volume percentage of red blood cells in blood. So it's the rate of the formed elements of blood, how much volume all the formed elements take in blood. And we have normal ranges for men, about 40–48% the formed elements make in blood, and in women lower volume, the 36–42%. If the hematocrit level is too low, it may be the result of anemia. If the hematocrit level is too high, it may indicate dehydration of the body, lack of water in the body. Blood plasma, which is extracellular matrix of blood as a tissue, includes water about 90%, and the rest 10% is proteins, electrolytes, which means ions, nutrients, amino acids, lipids, sugars, etc., metabolic products, including wastes, gases, and biologically active substances, such as hormones and others. Plasma proteins include albumins, globulins, and fibrinogen. Albumins make approximately 60% of blood plasma proteins, and here the list of their functions. They ensure oncotic pressure, colloid osmotic pressure of the plasma. It means that the presence of such proteins, of albumins inside the blood in blood plasma prevents leakage of water from blood vessels outside to surrounding tissues. So the watery blood always stays inside blood vessels, um, because this water is attached somehow to these albumins, and this is called colloid or osmotic and oncotic pressure. If we have the lack of albumins, it will cause the leakage of water from blood vessels into tissues, and this will cause edema. Next, albumins being then transport bilirubin, cholesterol, fatty acids, bile acid salts, and other substances, including pharmaceuticals, attached to these albumins. Also, albumins provide a source of amino acids for nutrition. Albumins, they are proteins, so they consist of amino acids, and when we have a lack of food, for example, as a result of starving, the albumins from blood will be taken by the cells of our body and broken down into amino acids, and amino acids will be used by the cells of our body. So albumins are the storage for amino acids. Next, globulins, which about 20–30% of plasma proteins. They are gamma globulins and alpha and beta globulins. Gamma, alpha, and beta are the Greek letters. So gamma globulins, or the same immunoglobulins, they are the proteins released by the so-called plasma cells, which perform the function of humoral immunity, and they are known as antibodies. Alpha and beta globulins take part in transporting of lipids, hormones, vitamins, and microelements. And third class of plasma proteins is fibrinogen, and it is a glycoprotein complex which is transformed into insoluble fibrin during blood coagulation. So it serves for blood coagulation and clotting. In blood plasma, we have some buffer systems which ensure the constant pH level, and the constant level of pH is 7.36, and these buffer systems include carbonate, phosphate, and protein system. And the blood plasma without fibrinogen is called serum. Next, formed elements of blood. You can see them all here. These are called leukocytes, or white blood cells. They are actually cells because they have nuclei. They include neutrophils, eosinophils, basophils, lymphocytes, and monocytes. The other two, platelets and erythrocytes, are not cells because they do not have nuclei, and they can be called postcellular structures, derivatives of cells. Formed elements of blood include erythrocytes, leukocytes, thrombocytes, and their count per 1 liter of blood is called blood formula, or hemogram, or complete blood count, CBC, or blood index. It's the total count of formed elements of every type per 1 liter. And these are the numbers that you should learn by heart. These numbers. So we have 3.9 till 5.5 trillion of erythrocytes per liter in men. In women, a little bit lower. Leukocytes are 4 to 9 billions per liter. Thrombocytes or blood platelets comprise about 180 till 320 billions per liter. You should remember these numbers. Here in this picture and in this one we have the scanning electron microscopy picture colorized with some computer programs, because the electron microscopy pictures are black and white, and here they are colorized artificially. These are red blood cells or erythrocytes. These are leukocytes or white blood cells. Erythrocyte, white blood cell, and this small is a platelet. Next, if you take blood from the patient and let it stay for a while, you will see that the erythrocytes and other formed elements of blood will go downwards, and above them you will start to see the blood plasma here. And if you measure the distance in millimeters, the red blood cells fall in 1 hour. It will be called the erythrocyte sedimentation rate, or ESR, or SED rate. And it reflects the speed of falling of erythrocytes, and it reflects the protein composition of the plasma and electric charge at the surface of erythrocytes. And we also have the normal ranges for this ESR, which is 1 to 10 millimeter per hour in men, and from 2 till 15 millimeters per hour in women. If this ESR is increased, we can say that we have inflammatory process, inflammation, or infectious or oncological disease. It may be cancer. Age-related changes in blood formula, total blood counts, is described in this table. So in newborns we have a little bit higher level of erythrocyte if we compare it to adult level. Adult level is here, 3.7 till 5.5 trillions per liter. In newborns a little bit higher than normal. Then 14 days, almost the same as in adults. Next, at 6 months of development in baby, baby will have lower amount of erythrocyte, and it will be called physiological anemia. Anemia means lower than normal amount of erythrocytes or hemoglobin inside erythrocytes. Physiologic means that it is not some disease. It's normal for baby 6 months old to have lower than in adult amount of erythrocytes. And then the number of erythrocytes start to grow up till adult levels. So the graph will look like this. At first a little bit higher than normal, then amount goes lower, and then returns to adult value. What about leukocytes? In newborns we have higher than in adult number of leukocytes, 10 to 30 in newborns, and 4 to 9 billions per liter in adults. And while the baby grows, the number of leukocytes slows down, goes down, and the graph will look like this. Now we will talk about each type of formed element of blood, starting from the major one, erythrocytes or red blood cells, or for short RBC. They can be called postcellular structures because they have no cell nucleus, or they can be called the cells without nucleus. Their lifespan is about 120 days up to 4 months. They are produced in red bone marrow and they die in spleen and liver. They transport gases, antibodies, toxins, and amino acids. This is how they look like in scanning electron microscopy. This picture was artificially colorized in computer program, and this is how they look like in the inside the blood capillary. So this is the lumen of blood capillary, and inside it we can see erythrocytes here, and this small is a platelet. And here how the erythrocytes look like in the blood smear, stained with hematoxylin and eosin. So these are erythrocytes. Most of erythrocytes are normocytes, which have this specific shape, biconcave shape, or discocytes, which have the thinner area in the middle. So this is why the middle area is stained lighter than the peripheral part, and this is well seen also in our blood smears. So every erythrocyte normally has the lighter area in the middle. And also normocyte should have the normal size, so its diameter should be 7.5 micrometer. And so these normocytes are about 75% of all erythrocyte in blood smear, and other 25% can be of different shape and size normally. So about 12.5% of red blood cells can be smaller than normal, smaller than 7 micrometers, like this, and they are called microcytes. Some of them are larger than normal, also about 12.5% normally. Some of them have different shape, not normal shape. For example, spherocyte. Sphero means a ball, and they are distinguished by lack of this lighter area in the middle. They are about 1%. About 6% are echinocytes with some spikes at their surface, and they are aging forms of red blood cells. Some of them can be like these, acanthocytes and sickle cell red blood cells. Such sickle cell can be found in the sickle cell disease only, so this is the really pathological condition. If a person has too high amount of the erythrocytes with abnormal size, it will be called anisocytosis. Too many erythrocytes with abnormal size. If too many erythrocytes have abnormal shape, it will be called poikilocytosis. Both conditions are the features of some disease. Now, the membrane of erythrocytes include such proteins. So here we have the membrane, cytoplasm inside. Cytoplasm consists of water and hemoglobin. No organelles are present inside the erythrocyte, just the hemoglobin dissolved in water. And this is the membrane of erythrocyte. Just below the membrane we have the specific elements of cytoskeletons specific for erythrocytes. It consists of protein called spectrin. It's a main cytoskeleton protein of red blood cell, and it consists of fibrils made of 2 chains, alpha and beta chains. And a spectrin network maintains the shape of erythrocyte and helps them to change their shape and to pass through very narrow capillaries. Next, we have the transmembrane proteins, Band3 protein and glycophorin protein. Both are transmembrane integral proteins. Band3 protein is actually the ion transporter, the chloride bicarbonate anion exchanger. It means that it allows one ion inside and other ion outside, and thus it provides the transportation of CO2 gas by red blood cell. Glycophorin is a receptor glycoprotein. It has the carbohydrate chains bind to its surface, and these carbohydrate chains have the negative charge, and they give the negative charge to all the outer surface of erythrocytes. So all erythrocytes have negative charge, and also the blood vessels also are negatively charged from inside, and thus it prevents the contact, the sticking of erythrocytes to each other and to walls of blood vessels from inside. And finally, we have the proteins which connect spectrin to transmembrane proteins, and these anchoring proteins are anchoring and band 4.1 and actin proteins. Anchoring binds spectrin to band 3. Band 4.1 and actin bind spectrin to glycophorin. In blood smear, we can find also so-called reticulocytes, which will look like these, with some so-called reticulofilamentous substance stained with basic stains such as methylene blue, and this reticulofilamentous substance is actually the not fully dead organelles, and they are present in premature, not fully mature red blood cell. So reticulocyte is not fully mature erythrocyte. They normally are up to 1% of all erythrocytes, but if their level is higher, it means that we have loss of blood, for example, as a result of trauma, some wound, or some other pathological condition. Next, thrombocytes or platelets. Here you can see how they look like in blood smear. They are about 2 to 4 micrometers. It means that they are much smaller than erythrocytes. Here they are. And they are postcellular structures. They are actually fragments, small pieces of cytoplasm of a giant cell called megakaryocyte, which is located in red bone marrow. So here, here, and here you can see the megakaryocyte, from which the small particles of cytoplasm is shed away in the form of thrombocytes, of platelets. So megakaryocyte and platelets are shed away from megakaryocyte. Another example here, and platelets are here. Function of platelets: they prevent bleeding by releasing vasoconstrictors, forming temporary plugs, and promoting blood coagulation. The structure of thrombocyte or a platelet include two areas. So in shape, each platelet is the disc, small oval disc, and each has two areas. The peripheral area here, it is called hyalomere, and internal area, which is called granulomere. Hyalomere includes so-called canalicular system with small channels and cytoskeleton elements. The canalicular system include open canalicular system, which is small intracellular channels, and closed canalicular system with some electron-dense content, which stores and release calcium ions and prostaglandin, which are required for blood coagulation. And second element of hyalomere are cytoskeletal elements, which are microtubules, microfilaments, and intermediate filaments. Microtubules are located at the periphery in the form of rings, and they maintain this disc shape of the thrombocyte. Microfilaments, actin, myosin, glycerine, and other are contractile proteins. It means that they can contract, and they are located peripherally between microtubules and membrane, and they participate in change of shape of platelets during their activation when they make the blood clot. And also they perform in platelet contraction and clot retraction. When the blood is stopped, when the damaged wall of blood vessel is healed and the blood clot is not needed anymore. Granulomere here, it includes some organelles such as mitochondria, ribosomes, small cistern of granular endoplasmic reticulum, rough endoplasmic reticulum, and Golgi apparatus, and also inclusions of glycogen. And second component of granulomere are granules. Granules of three types, labeled by Greek letters: alpha granules, delta granules, and lambda granules. So this is kind of top view, and this is cross-section of a platelet. So at the periphery we have the hyalomere with microtubules, very well seen here, and here they are in cross-section, and granulomere with granules and some organelles. What about granules? We have three types. Alpha granules, they contain blood clotting factors and proteins. It means that they serve for blood coagulation and blood clotting. These proteins are glycoproteins such as fibronectin, fibrinogen, and von Willebrand factor, and also thrombospondin. Next, delta granules contain ADP, ATP molecules, which give energy for all the chemical processes in platelets. Calcium ions also need for these processes, and also serotonin and histamine. These serve for changing of permeability of the blood vessel. And lambda granules contain thrombolytic enzymes. Thrombo means about the blood clot, lytic means for lysis, for breaking down. So these enzymes are needed when the blood clot is not needed anymore, and we should break down it to let the blood flow through the blood vessel. So these thrombolytic enzymes include acidic hydrolases, lipases, phosphorylases, phosphatases, and microperoxisomes. Here in this diagram you can see what happens when the blood vessel is damaged and what is the function of platelets. So here we have the damage of endothelium. Endothelium is the wall of blood vessel. So here the wall is damaged, the von Willebrand factor is released, and it causes adhesion, the attaching of thrombocytes from blood to the wall of the blood vessel here. Next happens the activation of platelets of thrombocyte and changing of their shape from discoid-like to this, like star-shaped with long processes. And this happens with the help of the elements of cytoskeleton and other molecules. So the components of granules are released, such as serotonin, ADP, other factors and proteins, and the clot is formed with the help of fibrinogen, and the aggregation happens and the clot is formed. Next, what will happen next? The endothelium will close this damaged area, and the wall of blood vessel will be restored, and then the blood clot which consists of thrombocytes and other formed elements of blood and fibrin. It is no needed anymore, and it should be broken down, and it is happening with the help of enzymes which are stored inside the platelets. So the clot will be broken down and the blood will move through this blood vessel. In this picture you can see all the internal structures of the platelets. This is the horizontal section and this is the cross section. So at the periphery we have the hyalomere with microtubules and actin and other microfilaments. In cross section here we have microtubules in cross section and here also, and at the periphery microfilaments. The membrane of thrombocytes is covered with carbohydrate chains with negative charge. So this is the glycocalyx, negatively charged. Inside we have the granulomere with some granules such as alpha granules, delta granules, and lambda granules, and other and organelles such as mitochondria, some elements of endoplasmic reticulum, and glycogen inclusions, and also two types of canalicular system. Canaliculi means small channels, open and closed one. Next we go to blood cells, which are called leukocytes or white blood cells. We have 5 types of them, and they are subdivided into 2 large groups: granulocytes and agranulocytes. These names come from what granule, it's about granules. So granulocytes have 2 specific features. They have segmented nucleus and specific granules. So granulocytes, neutrophil, eosinophil, basophil, have segmented nucleus consisting of several segments connected with very thin bridge. The same you can see here, segment 1, segment 2, connected with thin bridge. And specific granules, which are specifically stained and give specific name for these cells. So basophil has basophilic granules, eosinophil has eosinophilic granules, and neutrophil has both eosinophilic and basophilic granules. In turn, agranulocytes, A means no, so they have non-segmented nucleus, it's the whole nucleus, and no specific granules. And we have 2 types of agranulocytes, lymphocytes and monocytes. This is also what you should learn by heart. This is the composition of leukocytes. It is called leukocytic formula or white blood cell differential, and this is the percentage of leukocytes of every type among all the leukocytes. So among all the leukocytes we have 19 to 37% of lymphocytes from all the leukocytes, 3 to 11% of monocytes from all leukocytes, 48 till 78% of neutrophils, up to 5% of eosinophils, and up to 1% of basophils. And among neutrophils we have the neutrophils of different stages of differentiation. The most differentiated, fully differentiated, are called the neutrophils with segmented nuclei, or SEGs. They are about 47 up to 72% of all the leukocytes. Also, we can find the so-called bands. They are not fully mature neutrophils, up to 6% of all leukocytes, and so-called young neutrophils. They are less mature than bands. They are up to 0.5% of all the leukocytes. So this is the normal percentage of different leukocyte types in blood smear. If something changes, for example, if you see higher than normal amount of non-mature forms of neutrophils, it will show that we have the so-called left shift of the leukocytic formula. It means that we have somewhere some trauma, for example, or a wound, leakage of blood, and we need a lot of new formed blood, and this causes not fully mature, premature forms to exit the red bone marrow where they are born before they get fully mature. So it will be called left shift. Age-related changes in leukocytic formula from newborns till adults. Here we compare the counts of neutrophils and lymphocytes. Neutrophils serve for innate, non-specific immunity against bacteria, any bacteria, and lymphocytes are for specific immunity or adaptive immunity. So first, when the baby is born, he has high amount of neutrophils, about 65% of neutrophils and 25% of lymphocytes. Till at day 4, the amount of neutrophils and lymphocytes equals, and next at the 1-2 years we have lower amount of neutrophils and high amount of lymphocytes. Next at 4 years again we have equal amount of them, and these points are called physiological crossing, first and second physiological crossing. And by the adult age we will have the higher amount of neutrophils, 65%, and lower amount of lymphocytes, 25%. Here you can see the ultrastructure of granulocytes. You should be able to distinguish between different cell types according to their internal morphology. So here you can see the structural features of granulocytes. All granulocytes have the same features. They all have segmented nuclei and specific granules in cytoplasm. in picture A, we have the neutrophil. It has the higher amount of segments, 3 to 5 segments. The so-called bar body or drumstick. It is the second X chromosome in women. And neutrophil has small specific granules. Its size is 10 to 20 to 12 micrometers. Next, eosinophil. It has only 2 segments, one second segment of nucleus connected with small bridge, and very specific granules, specifically looking. They are oval with the so-called crystalloid in the middle. It is the condensed specific proteins released by eosinophils. So this is called the crystalloid inside specific granules. And basophil has also 2 segments of nucleus with small bridge and large specific granules. This is how the granulocytes look like in the blood smear. Here we have the basophil. It has a lot of basophilic granules, and because the nucleus is also basophilically stained, we cannot see cell nucleus because of a lot of granules. Too much granules, basophilic, to see the nucleus. Next, eosinophils in these 2 pictures, they have 2 segments of nucleus and eosinophilic granules in cytoplasm. And in lower 3 pictures we have neutrophils with 1, 2, 3, 4 or 5 segments of nucleus with bar body here labeled with the arrow, and more transparent, pale stained cytoplasm with very tiny red and blue granules. Structural features of agranulocytes. We have 2 types of agranulocytes. Lymphocyte and monocyte. Lymphocytes are the smallest cells in blood. They have rounded cell nuclei and very tiny cytoplasm. And they are subdivided into 3 subtypes according to their size: small, up to 7 micrometers, medium, 8 to 10 micrometers, and large, more than 11 micrometers. Monocytes are the largest cells. Their size is up to 20 micrometers, and they have the bean-shaped nucleus or kidney-shaped nucleus and a lot of cytoplasm. All the leukocytes, both granulocytes and agranulocytes, have pseudopodia which allow them to move. So all leukocytes can move and go wherever they want to perform their functions. Here we see the agranulocytes in blood smear. Here, small lymphocyte. It has so tiny cytoplasm that it is almost not seen. We see just its nucleus. And here we have large lymphocyte. It is larger than surrounding erythrocytes, but also nucleus is rounded in shape and the cytoplasm is very small. And here monocyte, largest cell with the bean-shaped nucleus and large cytoplasm. And now we will describe all these types one by one. First, neutrophils. They have from 3 to 5 segments of the nucleus with bar body in women, very small eosinophilic and basophilic granules in cytoplasm, and they have specific granules which contain specific proteins which serve for the functions of neutrophils, and the function of neutrophils is to kill and phagocytize bacteria. So their specific granules contain lysozyme, which destroys bacterial cell walls, lactoferrin, which sticks the bacteria, complement activators and destructive enzymes such as collagenase, phospholipase. The enzyme which ends on ase serves to break down what is before ase. So collagenase will break down collagen. Phospholipase will break down phospholipids. So collagenase help to get through the basement membrane of the blood vessel. In basement membrane, as you remember, we have collagen type 4 in lamina densa, and this collagen is broken down with the help of collagenase. Phospholipase destroys the phospholipid molecules of the membrane, and it breaks the membrane of bacteria. Here in the transmissional electronograms, you can see neutrophil here and here. In this one, in left picture, you can see the bridge between two segments and a very small specific granules in cytoplasm. But in picture on the right, you cannot see the bridges between the segments of the nucleus because of the plane of sectioning, but the granules are still seen. And what else should I say? That both granulocytes and agranulocytes both have the so-called non-specific granules. Non-specific granules are lysosomes. They all have lysosomes. Next, eosinophils. They have two segments of nucleus and eosinophilic specific granules. Non-specific granules are lysosomes. Eosinophilic granules contain proteins and other substances for two major functions of eosinophils. First function is antiparasitic function. Antiparasitic means against unicellular, such as amoeba, infusorium, and other unicellular parasites, and multicellular, such as worms. So antiparasitic function is first, and second function is the histamine inactivation during inflammation and allergic reactions. Histamine is released by basophils, but eosinophils break histamine down during these reactions. So specific granules of eosinophils contain substances for these two functions, such as major basic protein, eosinophilic cationic protein, eosinophilic derived neurotoxin, peroxidase, and lysosomal hydrolytic enzymes. They are for antiparasitic function. They destroy parasites. And histaminase breaks down histamine. This is for the second function, to inactivate histamine. In these pictures, transmissional electron microscopy pictures, you can see nuclei. Here the bridge is seen, in the second bridge is not seen between the segments of nucleus, but you can easily define this type of blood cell because of the specific crystalloid inside the oval granules. So you have here the oval granules with crystalloid inside. Next, basophils. Basophils also have two segments in nucleus, specific basophilic granules, which can be blue, purple, or dark red in color because of the presence of the heparin, which switches the color of the basophilic staining. And the basophils produce some biologically active substances such as heparin, histamine, prostaglandins, leukotrienes, and serotonin. And the degranulation or release of these granules causes the allergic reaction type 1 and stimulates blood flow, increase vascular permeability, and makes it easier for other granulocytes to come from blood vessel to the surrounding tissue in the area of the injury. And this is how basophil looks like in the electron microscopy picture. Segments of nucleus here. No bridges are seen in this particular picture, and these are large granules of basophil. In this picture, you can see the bridge between segments and large granules. Monocyte. Monocyte is the largest blood cell. Here is it. It has the non-segmented bean-shaped nucleus or kidney-shaped nucleus, a lot of cytoplasm with lysosomes in it. Actually, monocyte is not a final form of this type of cell, and when it goes out from the blood vessel to surrounding tissue, it will develop next into its final form. It may become macrophage or osteoclast or dendritic cell. Macrophages are the most numerous derivatives of monocytes, and they participate in specific and non-specific immune reactions. They can perform phagocytosis of anything, of bacteria, of parasites, of dead cells, of extracellular matrix, etc. And also they can perform the function of so-called antigen presentation. It is the part of immune reaction. Also, macrophages can release antibacterial substances and inflammatory signaling molecules to activate other leukocytes during inflammation. And also, macrophages can promote tissue repair. Here you can see the electron microscopy pictures. Monocytes, very large cells with a lot of cytoplasm, with a lot of vacuoles and lysosomes. Here also, and bean-shaped nucleus. Monocytes have a lot of lysosomes because when they will convert into final form, into macrophage, it will perform a lot of phagocytosis, and during phagocytosis, the engulfed particle, the eaten bacteria, for example, or eaten cell, dead cell, should be digested inside the cytoplasm of macrophage with the help of lysosomes. So, monocyte has a lot of lysosomes for performing the function of phagocytosis when it will become the macrophage. Lymphocytes are the smallest blood cells. Here you can see the small lymphocytes, and this one is large lymphocyte. They have non-segmented round nucleus, a very thin cytoplasm with some amount of lysosomes. Also you can see here round nucleus and tiny cytoplasm with some organelles and some lysosomes. Morphologically, they are subdivided into three subpopulations: small, medium, and large. And functionally, according to their functions, they are subdivided into T, B lymphocytes, and NK, natural killer cells. Functions. Functions depend on the subtype of lymphocytes. NC cells are the cells of non-specific immune defense. B cells perform specific immunity, the so-called humoral immunity. They release antibodies or immunoglobulin, other name for it. The T cells, cytotoxic T cells, they also perform specific immunity, especially cell-mediated immunity. They kill genetically compromised cells, such as transplanted cells, the cells which were infected with viruses, or cancer cells. All these transplanted, infected with viruses and cancer cells differ from normal cells in their DNA. For example, infected with virus cells will have the DNA of virus, and they will produce the proteins of virus. Helper T cells, they mediate, stimulate specific humoral and cell-mediated immune reactions, and regulatory T cells suppress immune reactions. So here you can see the lymphocyte populations. Lymphocytes are subdivided into T, B, and zero lymphocytes. Zero lymphocytes develop into natural killer cells. B lymphocytes develop finally into plasma cells, which release antibodies, and memory B cells, which store the information about the infection. T lymphocytes are subdivided into helper T cells, cytotoxic T cells, suppressor T cells, and memory T cells. And here you can see the percentage of different types of lymphocytes in blood. Immunity is the ability of organism to protect its genetic identity. It means that any cell that has changed DNA should be killed. Antigen is any substance which is perceived as foreign by cells of immune system. So the virus can be antigen, bacterium can be antigen, anything which is not normal for our body, any protein which is not normal for our body can be seen by our immune system as an antigen. Immunity is subdivided into innate or non-specific and adaptive specific. Innate non-specific is performed without any specific receptors. It is performed with macrophages and granulocytes. Granulocytes are neutrophils, eosinophils, and basophils. These cells, macrophages and granulocytes, will attack and kill any antigen, bacterium, parasite, no matter which type of bacteria it is. So this is why it is called non-specific. Adaptive or specific immunity is performed with the help of specific receptors, and these are receptors for specific antigens on T and B cells. And in this case, every T and B cell has its specific receptor for only one type of bacterium or virus, and each T or B lymphocyte can attack only this one particular specific bacterium and not other ones. So this is why this type of immunity is called specific. And this immunity is subdivided into cell-mediated and humoral. Cell-mediated immunity is performed against cells, against infected with viruses cells, against cancer cells or transplanted cells, and it is carried out with the help of effector cells, cytotoxic T cells, which kill these bad cells. Humoral immunity is against substances, against antigens, and it is performed with the help of the effector cells, plasma cells. Plasma cells is the final form of B lymphocyte. So cell-mediated immunity with the help of T cells, cytotoxic T cells, and humoral-mediated immunity with B cells, plasma cells. So here is just a description of humoral and cell-mediated immunity once again. Once again, we have three populations of lymphocytes: T lymphocytes, helper, cytotoxic, suppressor, and memory T cells. B lymphocytes have two final forms, plasma cells and memory cells, and zero lymphocytes are natural killer cells. And here are their specific functions. Natural killers perform destruction of self cells that cannot be killed by cytotoxic T cells. If T cells fail to kill some bad cell, for example, some cancer tumor cell, it may be killed by natural killer cell. Memory T and B cells provide a rapid immune response if this type of antigen enters our body once again. And this is how vaccines work. Cytotoxic T cells destroy virus-infected tumor cells and perform cell-mediated reactions. Helper T cells activate such reactions. Suppressor T cells dysregulate these reactions. Plasma cells, final form of B lymphocytes, produce antibodies and perform humoral immunity reactions. In this picture, you can see the blood smear, in which you see red blood cells here, normocytes. This one is microcyte, smaller than normal. This looks like echinocyte with small spikes on its surface. Also, these small are platelets, here and here and here, also platelets. And this one is a neutrophil, because it has 1, 2, 3 segments connected with thin bridges and pale cytoplasm with very tiny granules in it. In the picture below left, we have the lymphocyte. This one is the medium and this one is large lymphocyte because of round non-segmented nucleus. And this one is a monocyte because it is very large if we compare it with erythrocytes, and it has the specific bean-shaped nucleus and a lot of cytoplasm. Here in the left picture we have a lymphocyte, round non-segmented nucleus and small amount of cytoplasm. Here this one looks like basophil because it has basophilic granules, so multiple that we cannot see nucleus covered with these granules. This one is neutrophil with 1, 2, 3 segments and pale cytoplasm with tiny granules. In the picture below we have once again a neutrophil because it has pale transparent cytoplasm and nucleus with 1, 2, 3, 4 segments. And at the right hand from this neutrophil we have the eosinophil with eosinophilic red granules. In the picture on the right we have a neutrophil with pale cytoplasm, 1, 2, 3, 4 segments, and a so-called drumstick or bar body, which is present only in women. Here you can see the table with the most important functions of all the formed elements of blood. So this is a very important table for you. You should know it by heart. And now we go to hemopoiesis. Hemo means blood, poiesis means creating. So hemopoiesis is the process of production of blood. And it is subdivided into processes of embryonic hemopoiesis, when the blood is first formed as a tissue, and post-embryonic hemopoiesis is the renewal of blood elements. Here you can see the embryonic and fetal hemopoiesis. It has 3 phases. First, mesoblastic. It happens up to 3 months of embryonic development in the wall of the yolk sac, outside the embryo itself, in the temporary extra embryonic organ in yolk sac. Next, this process goes into liver and spleen, and now it is called hepatic phase of the hematopoiesis. Hepatic stands for liver because most of the cells now are produced in liver, cells of blood I mean. And next, when the bones of embryo start to be formed, then this process of hematopoiesis goes to bone marrow when it first appears. And up to birth, all this process of hematopoiesis now goes inside bone marrow, which is located inside the bones. So by the birth, all hematopoiesis goes, most of the hematopoiesis goes inside the bone marrow in different bones, and then during aging, the rate of hematopoiesis slows down, and for example, in long bones, first it goes in both diaphysis and epiphysis, but then in diaphysis, the red bone marrow is substituted with yellow bone marrow, which is just adipose tissue, the fat tissue, and it stays only in epiphysis of the bones. During embryonic development, the hematopoiesis starts in the wall of yolk sac. So this is the sagittal section of embryo. Embryo itself is here, embryonic disc. Above embryo we have amnion, below embryo we have yolk sac, and in the wall of yolk sac we will have the first blood vessels with first blood cells. Here we have extraembryonic mesoderm, which is also called extraembryonic mesenchyme, which looks like this, the cells with a lot of processes, and then they start to come closer to each other, and they become the first cells of blood, primary erythroblasts, and some of mesenchymal cells become the cells of blood vessels, endothelial cells. And this is how it looks like in the embryo section. First erythrocytes, they have cell nuclei first, and the wall of blood vessel is formed. Next, the process of the hematopoiesis goes into embryo itself. So first blood vessels and first blood is formed outside the embryo, and only after in the embryo itself, while it develops. So here, in the cross-section of the chicken embryo, here you have the aorta sections. In it you can see the blood cells, primary blood cells, and also in this area between the visceral layer of splanchnoderm and endoderm, between these two layers we have also mesenchyme, in which you can see these round or oval in shaped cross sections of blood vessels. At high magnification, one of such blood vessels, and inside it you can see primary blood cells. During postnatal period we have the hematopoiesis mainly in red bone marrow, inside these small cavities in bones. The structure of hematopoietic differon include these steps. The first step, the first cell from which all the formed elements of blood are developed, is called hematopoietic stem cell. HSC stands for hematopoietic stem cell. Next goes multipotent progenitor cell, next unipotent precursor, then blast, then maturing, and then mature specialized cell. First hematopoietic stem cell is the pluripotent cell. It means that it has very many ways, potential ways of development. It can become any formed element of blood. Then it becomes multipotent progenitor, which can become not all the formed elements of blood, but only some of them. So it has fewer potency. Then unipotent. Uni means one, potent means potencies, potential ways of development. A unipotent precursor can become only one formed element of blood. For example, only neutrophil, not erythrocyte, not monocyte. Then blast. Blast can divide very fast and very quickly. Maturing cell loses ability to divide and becomes gradually specialized cell. And specialized cell is fully mature cell. It goes out from red bone marrow into blood vessels, into blood circulation. This is how this table looks like. Another example of this table, hemopoiesis or hematopoiesis, is subdivided into 6 classes. Here we have cells of 6 classes. Class 1 is hematopoietic stem cell. It is pluripotent. It can become any of blood-formed elements. Then it divides and can become either this one, multipotent progenitor, or this one. These are the cells of class 2, multipotent progenitor cells. This is the colony forming unit. CFU stands for colony forming unit, because if you plate it into the laboratory plate, it will divide and make the colony of the same cells. So it is called colony forming element. And it is the colony forming element for granulocytes, erythrocytes, monocytes, and megakaryocytes. So for short, GEMM. G for granulocytes, M for monocytes and megakaryocytes, and E for erythrocytes. Then class 3 is the committed oligopotent and unipotent precursors, colony forming units. They are already unipotent. Each of them can divide only into one formed element of blood. So this one is called, for example, colony forming unit for monocytes. This one colony forming unit for neutrophils, etc. Next, class 4 are blasts. They are called blasts. They are precursors of particular blood lineage, and they are larger than the cells of class 3, 2, and 1, and have larger nucleus with a lot of euchromatin, because they constantly divide and make more such cells. They are all called blasts. Monoblasts, neutrophilic myeloblast, megakaryoblast, erythroblast, T lymphoblast, etc. Next, class 5. They are differentiating cells, and when they differentiate, they gradually lose their ability to divide. So the cells here still can divide, and here lose their ability to divide. And they gradually gain some specific features. For example, granulocytes in class 5 gain first non-specific granules, which are lysosomes, and then specific granules, specific for each type of granulocytes. And finally, class 6, post-mitotic cells. It means that they cannot divide via mitosis, and they are mature cells that can go out from red bone marrow to systemic circulation. What can we say about particular differons. So differon is the cell type, one type of formed element of blood starting from hematopoietic stem cell. For example, you should be able to build up, to name all the components of any differon. For example, the task is to name all the components of differon of erythrocytes. It means that you should name first hematopoietic stem cell, then colony-forming unit of granulocytes, erythrocytes, monocytes, and megakaryocytes, then burst-forming unit of erythrocytes, then colony-forming unit of erythrocytes, then erythroblast, then basophilic proerythrocyte, then polychromatophilic proerythrocyte, then oxyphilic proerythrocyte, then reticulocyte, then erythrocyte. I have named all the components of differon of erythrocytes, the same as for any other of them. For example, for neutrophils, it will be hematopoietic stem cell, colony-forming unit of granulocytes, erythrocytes, monocytes, and megakaryocytes, then colony-forming unit of monocytes and granulocytes. Next, colony-forming unit of neutrophils, neutrophilic myeloblast, neutrophilic promyelocyte, neutrophilic myelocyte, neutrophilic metamyelocyte, neutrophil stab cell, neutrophilic granulocyte or SEG. You should be able to name all these cells and build up any cell differon. And also you should be able to explain what happens during differentiation of any cell in blood. For example, if I ask you what happens during erythropoiesis, you say that first hematopoietic stem cell becomes from pluripotent multipotent and loses ability to become lymphocyte. So this cell cannot become lymphocyte, only some of these cells. Then it becomes unipotent precursor of erythrocytes, and during this development in these three stages, all that happens inside nucleus. Inside nucleus, some genes are blocked. For example, when this cell becomes this cell, the genes for development into granulocytes and monocytes and megakaryocytes are blocked, and only genes for becoming erythrocytes stay active. So during first stages, everything happens inside nucleus with genes. Some of them become blocked. Next, during stage of erythroblasts divide and proliferate a lot. Then erythroblast becomes basophilic proerythrocyte. Basophilic because of the color of cytoplasm. In cytoplasm, a lot of polyribosomes develop, which serve to produce hemoglobin, which in the end should fill all the cytoplasm of erythrocyte. So polyribosomes make the cytoplasm basophilic and give this name to this cell, basophilic proerythrocyte. Then polyribosomes start to produce hemoglobin, which is eosinophilic, and eosinophilic plus basophilic color makes a violet purple color and gives the name polychromatophilic proerythrocyte. Then next stage, all the needed hemoglobin is now synthesized, and polyribosomes are not needed anymore, and they are degenerating. So no polyribosomes, no blue color, only hemoglobin with red color. And this is why this cell has oxyphilic cytoplasm, which gives the name to the cell, oxyphilic proerythrocyte. Next, the cell nucleus goes out from the cell, And we have reticulocyte and then erythrocyte. What happens the same during differentiation of any of granulocytes? For example, if we talk about eosinophils, so the same during first three stages, some genes are blocked, and the unipotent precursor colony-forming unit of eosinophils can then develop only into eosinophils, not into any other types of cells. Eosinophilic myeloblast divides a lot, and then during class 5, specific features start to be seen. First, in the stage of eosinophilic promyelocyte, the primary non-specific granules develop. Primary non-specific granules are present in all leukocytes. They are lysosomes. They are basophilic. Then, on the stage of eosinophilic myelocyte, in addition to non-specific granules, specific granules are born in the cytoplasm of the eosinophil, and the specific granules of eosinophil are eosinophilic. They are red in color. Then, during next stages, the eosinophilic metamyelocyte loses its ability to divide. It cannot divide anymore. Its nucleus becomes smaller and darker. It is condensed, so we can see here the pycnosis of nucleus, and its shape changes from round to this segmented. Eosinophilic stub cell has curved nucleus, and finally eosinophilic granulocyte is the cell with two segments of nucleus connected with thin bridge. And this is the post-mitotic class 6 cell which can exit red bone marrow and go to blood. During the development of platelets, what happens? The same for classes 1, 2, and 3. Next, megakaryoblast, the cell which divides a lot, and then class 5, it gains specific feature. Megakaryoblast develops into pro-megakaryocyte and next into megakaryocyte. Mega means very large. Karyo stands for nucleus, cyte is cell, so the name means that this is a giant cell with a giant, very large nucleus. It is a result of endomitosis, during which the cell becomes not diploid but tetra and multiploid, such as 64N. So it has a lot of DNA and a very large nucleus and very large cytoplasm. And as you can see here, the pieces of cytoplasm of the mature megakaryocyte start to detach, to shed away and become platelets, because platelets are the pieces, fragments of cytoplasm of megakaryocyte. And during development of pro-megakaryocyte and megakaryocyte, what happens in cytoplasm, which will next become platelets? Everything that we see finally in platelets start to develop. So, as we remember, in platelets we have elements of cytoskeleton at the periphery and granules of three types in the middle and some organelles. So this all starts to develop in this stage, in pro-megakaryocyte, and it is present in the cytoplasm of megakaryocyte, and pieces of the cytoplasm are detaching and become platelets with all these components such as granules, cytoskeleton elements, etc. What about lymphocytes now? Lymphocytes do not develop any specific morphological features because everything happens inside their nucleus first and upon their membrane second. Their differentiation is the development of specific receptors upon their surface. Here in this picture we can see only T and B lymphocytes, but we know that we should have here the third line for 0 lymphocytes or natural killer cells. Okay, we have here only T and B cells. T cells are for cell-mediated immunity and B cells for humoral immunity. T cells will have specific T cell receptors for antigens, and B cells will have B cells receptors. The T cells develop first inside red bone marrow, and then from class 3 they go out from red bone marrow and go to thymus, where they complete their development. But these cells of 6th class, T lymphocyte and B lymphocyte, are not their final forms. They exit red bone marrow, B lymphocyte and T lymphocytes exit thymus when they are the cells of class 6, and then they go to peripheral organs of immune defense, such as lymph nodes, spleen, and tonsils, where they complete their differentiation into their final forms. The final form of T lymphocyte will be activated cytotoxic T cell, which kills cells infected with viruses and cancer cells, and final form of B lymphocyte will be a plasma cell, which produces antibodies or immunoglobulin, the same, which will act against antigens. What about the stem cell, hematopoietic or hemopoietic stem cell? It looks like a small lymphocyte, has high nucleus to cytoplasm ratio. It means it has very small cytoplasm. It is unique, cannot be substituted by other undifferentiated cells from other tissues. It is capable of self-renewal and differentiation. It can divide with asymmetric mitosis. It means that one cell stays in undifferentiated stage, and second cell goes to differentiation process. They are their divisions are very, very rare, so they are at resting stage. They are pluripotent, and they have specific microenvironment. So they are surrounded with other cells which produce some specific substances that help the stem cell to be the stem cell, not differentiated one, because we want to have stem cells for all our lifespan to make all the kinds of formed elements of blood. Because if all the stem cells will at once differentiate, we will not have any more other stem cells. We want to have some pool of non-differentiated stem cells always. So the surrounding cells release some specific substances to maintain the pool of non-differentiated stem cells. So about the T and B cells, as I said previously, B cells continue their development inside red bone marrow, but T cells from class 3 go out from red bone marrow to thymus, and there they develop up to class 6. And then both T and B cells go to peripheral organs of immune defense, spleen, lymph nodes, and tonsils, and complete their differentiation. Monocytes develop into macrophages and also some dendritic cells and microglial cells. Macrophages perform the function of phagocytosis, so they eat anything. They can eat bacteria, other cells, the components of extracellular matrix, anything. Once again about T lymphocytes, and finally some pictures showing our histological preparations in which you should be able to define any formed elements of blood. So the preparation is called blood smear. it is stained with azure eosin stain, not hematoxylin, but azure instead of hematoxylin. Azure is also basic stain as hematoxylin is, but azure is usually used for exactly blood smears. So the staining is called azure eosin. What do we see here? We can see here a lot of red blood cells or erythrocytes. They are normocytes because we can see here the lighter area in the middle. It means that they have the normal shape as discs. These small particles, they are thrombocytes or platelets. And these are the cells with nuclei. It means that these are leukocytes. This one can be a monocyte because it is very large. It has large nucleus and a lot of cytoplasm. This one should be a neutrophil because it has 1 to 3 segments of nucleus. This one is a small lymphocyte with tiny cytoplasm not seen and a small round dark nucleus. And this one again monocyte, large cell with the cell nucleus with irregular shape and a lot of cytoplasm. In this picture, what can we see? This is the stub cell. This is not fully mature neutrophil. This one also should be the stub cell, and this one a lymphocyte. And these are erythrocytes. These are platelets. This is the aging form of erythrocyte. This is the echinocyte with small projections of the cytoplasm. It is the result of breaking down of the spectrin molecules which make the cytoskeleton of erythrocyte, which help erythrocyte to maintain its shape. And when this cytoskeleton is broken down, the shape changes, and this is what we see here. So this is the dying erythrocyte, and it will be soon destroyed inside spleen. In this picture, a lot of erythrocytes again. This one is a lymphocyte because it is a small cell with round cell nucleus and very small cytoplasm. This one not very well seen, but it should be the eosinophil because of the color of granules. If the color of granules looks the same as the color of erythrocyte, then we can say that this one is eosinophil. It is not very well seen in this particular picture because maybe this one is the basophil. If you are asked about the color of granules, you should answer that you compare the color with the color of erythrocytes. Erythrocytes are always stained eosinophilic. So if the color of granules is the same as the color of erythrocyte, then you say that this is the eosinophil. If you think that granules are more basophilic, then you say that this is a basophil. I'm not sure about this particular picture. Here in this picture, this is absolutely basophil because it has basophilic granules and a nucleus with 2 segments. This one is a mature form of a neutrophil with 1 to 3 segments with small bridge and pale cytoplasm. Another example of the same cell, neutrophil with 1 to 3 or 4 segments and pale cytoplasm with tiny eosinophilic and basophilic specific granules. Another example of a neutrophil. Neutrophil make up to 78% of all leukocytes, so they are present almost in all the fields of view. If you see the blood smear, they are much more frequently seen as other types of leukocytes. Another example of a neutrophil, this one is a basophil because it has two segments of nucleus and basophilic granules. These are platelets. This one is an eosinophil. It has two segments with small bridge and eosinophilic granules. This one again, the eosinophil, because of the color of granules, the same as the color of erythrocytes, and two segments of the nucleus. This one is a lymphocyte because it has non-segmented round nucleus and small amount of cytoplasm, so it can be the medium lymphocyte. Another example of the same cell, also medium or small lymphocyte because it has non-segmented round cell nucleus and tiny cytoplasm. Another example of lymphocyte, but this one can be medium or large lymphocyte because it is larger, but again it has round nucleus and small cytoplasm. This one is a monocyte because it is very large. If you compare it to diameter of red blood cells, it has the nucleus of irregular shape and a lot of cytoplasm. Cytoplasm is pale with no eosinophilic or basophilic granules, only lysosomes are present in monocytes. And this one is a lymphocyte. This one is a small lymphocyte. This one again a monocyte because it is very large. The nucleus is bean-shaped or kidney-shaped and large cytoplasm. It is pale or slightly basophilic because of lysosomes. Another example of a monocyte, once again because it is very large, bean or irregular shape of nucleus and large, lightly bluish cytoplasm. Erythrocytes, thrombocytes, and aging form of erythrocyte, echinocyte. During quiz, you should be able to name the differon, all the components of the differon, and the characterization of the process. It means that, for example, I show you this picture and I ask you which differon is shown here. Your answer should be that this is the differon of erythrocytes. Then you should name all the cells that are present here, name the cell that should be here in this frame. For example, this is the erythroblast, then here should be basophilic proerythrocyte, this is polychromatophilic proerythrocyte, this one is the oxyphilic proerythrocyte, reticulocyte, and erythrocyte. You should name them all. You should say which class they are. For example, erythroblast is a cell of class 4, and all others up to erythrocytes are the cells of class 5. You should be able to describe what happens during differentiation, such as change of color of cytoplasm and what happens to nucleus, etc. The same here. Name the differon, the classes, and the process. So, this is about the megakaryocytes and production of platelets. So, here is the megakaryoblast, promegakaryocyte, and megakaryocyte from which platelets are detached. And the same is here. So in this frame should be promegakaryocyte, megakaryocyte, and platelets. And the same for other cells. So it means that you should learn the table of hematopoiesis by heart. I mean this table, because you should be able to name all the components of any differons here. So that's all for now. If you have any questions, I will be glad to answer them. If not, then I wish you to have a good day, and goodbye to everyone.